seminar. We continue with the topic of COVID-19. Just a few words about Professor René Rossi. He is the head of Empa's Laboratory for Biomimetic Membranes and Textiles since 2003, and he holds lecturing appointments at ETH Zurich and also at the Université de Haute-Alsace in Mulhouse. He is also a member of the famous COVID-19 Swiss task force now. So I had opportunity to meet Professor Rossi a couple of years ago during the preparation of a project aimed at studying the potential of stormwater systems to mitigate urban heat events and improve human thermal comfort. Unfortunately, this project was not funded, and we'll look forward to find new opportunities to collaborate. But today, the presentation will not focus on this hot topic, I would say. It's about another hot topic, and we'll focus on the extensive expertise of Professor Rossi on the development of new materials and systems for the protection and optimal performance of the human body. More specifically, I think today we'll learn about the development of new membranes for protective face masks, and hopefully also trigger the discussion about the possible application of these new membrane materials for water and wastewater treatment. So after this small introduction, it is now time to listen to Professor Rossi. So the virtual floor is now yours. Thank you very much for joining the Eawag seminar.
Okay, thank you very much, Joao, for the kind introduction, and thank you for the invitation to present my work at this Eawag seminar. We are neighbors, or virtual neighbors in Dübendorf, but actually I'm working on the site in St. Gallen. So as you said, I'm heading a laboratory on the development of membranes and textiles, and we are particularly interested on applications around or in the body. So these applications can be sensors like this photo that we see here. So these are optical fibers that we develop ourselves that have a very high flexibility so that they are comfortable to wear. And with these, we monitor some vital functions of the human body. So in this case, this is a development with the Department of Neonatology of the University Hospital of Zurich, where we monitor the blood oxygenation of neonates. And here another example or schematic of what we can do. So we combine different materials into hybridic structures or multi-material structures for instance, here in this example for drug delivery applications. And we use some smart polymers so that we can trigger the response on demand, for instance, for better wound healing. And lately, I've been quite busy with the work in the science task force, and that's also the topic of my presentation today. So I divided the presentation into 3 topics. So first, probably this work is already quite well known, but I will summarize the work that we have in the group of infection prevention and control. We are very intensively discussing the needs for masks, and also we released some documents on clarification on what is the best use and how to handle the mask. But at the beginning of the pandemic, we also released a document that were recommendations for the quality of so-called community masks, so textile-based masks that as many industrial partners, so from the textile industry, had saw the need to start producing these sort of textile-based masks. And then in parallel, we started a large Innosuisse project that where we currently have around 50 partners. And here I will present some of the developments that we have recently been doing with the industry in the last few months. And from there, as it's also part of the development of the masks, I will go a bit more into details with the development of smart membranes. So about the mask types, before the pandemic in Europe, we only knew 2 types of masks, the FFP masks and the surgical masks. Both are have to comply with the requirements of European standards. So for FFP, we have EN 149, and the quality of these masks is defined in ordinances and in national regulations. So in Switzerland, we have this so-called PSA-Verordnung, which is the ordinance for personal protective equipment. So these FFP masks are protective equipment. And depending on the level of filtration efficiency, we have these 3 types of masks, FFP1, 2, or 3. So these masks corresponds to the American standard N95 or the Chinese standard KN95. So these denominations are not used in Europe, but they are frequently still used in the newspapers. Then the surgical masks, they correspond to another ordinance, that is the Medizinprodukteverordnung, the ordinance for medical products. They also, the surgical masks also correspond to European standard, so EN 14683. And here again, we also have 3 types of masks, so type 1, type 2, and type 3, depending on their filtration efficiency and depending on their air permeability. For the FFP masks, we have a filtration efficiency that is measured with small particles, so the particles in the order of magnitude of nanometers. And for the surgical masks, we only have a filtration efficiency against bacteria. And that's something that many people are mixing up when comparing the performance of different types of masks. We also have different test methods that are used. So basically, the surgical masks are not tested against aerosols. They're testing against these bacteria. These are Staphylococcus aureus. They have a size of around 1 micrometer, but usually they're aggregated in more or less bigger agglomerates of around 3 micrometers. So this is important to understand the filtration efficiency of these masks. What we also have to know is at the beginning of the pandemic, with the COVID ordinance that the Federal Council released, it was possible to sell other types of masks that did not correspond to a standard. So therefore, we still have in Switzerland some masks that look very like these surgical masks, but are denominated as non-medical masks. So they look very similar, but they do not have this certification according to the European standard. And so to more or less clarify the quality of these non-medical masks, we released in the Science Task Force this recommendation for a minimal quality of these textile-based masks. And in March, April, we worked very closely with different other countries, especially with France. That was the first country, apart from Switzerland, that also published some specifications for these textile-based masks. And this is this document here of AFNOR. So AFNOR, that's the French standardization body. And France was very active, so they also pushed their national document to the European level. And now we have a European document here, this SEN document. It's a SEN workshop agreement. It's not a standard, but that's a document that is used to classify the quality of these community masks. So basically, we have 2 documents that can be used in Switzerland for the quality of these community masks. On one hand, the recommendation of the Science Task Force, or this French or European document that is especially also used in the French part of Switzerland. So here is the group that worked on the development here of these recommendations. So apart from material science institutes like Empa and the 2 ETH, we also have in this group a large number of hospital partners. So from Zurich, Bern, Basel, Geneva, Winterthur, we have these different partners that help developing these recommendations. And this also helps understanding the requirements that we set. They are really very near from the requirements of the surgical masks standards. So basically, what we looked first is what could be the problem. So we looked at which is the size of droplets that are released when speaking or breathing or coughing. So we see here from this rather old study, but it's still used quite intensively here, the distribution here of the droplet size. And we see that we have a peak around 1 micrometer, but especially when speaking or singing, we also produce droplets that have much larger sizes, so that can go up to even higher than 10 micrometers. And therefore, we decided in the Swiss document to look at the filtration efficiency with a particle size here of 1 micrometer. And that's a big difference that we have compared to the French or European document. They look at the filtration efficiency with particle sizes of 3 micrometers. And we think, looking at now the more and more intense discussion about the effect of aerosols on the contaminations, we think that it was the right choice to choose here this particle size of 1 micrometer. And so we defined with these textile-based masks a filtration efficiency of higher than or equal to 70%. Then, as we knew that also some large droplets are produced, we took over more or less a test that is used in the standard for surgical masks. That's usually a protection against blood projections, and we adapted this test. So we reduced the pressure that we use to project here this jet. We reduced it to 12 kilopascal. That corresponds to the pressure that where let's say droplets can be released when coughing. And we defined that no liquid penetration should occur in these masks. And then the third, so this test is not required in the French or European documents. So they really concentrated on filtration efficiency and air permeability, because the French document was more directed towards the FFP standard. And in Switzerland, as I said, we looked more at the standard on surgical masks. So we have a certain air permeability also. So here we look at the air resistance. So here it should be lower than a certain pressure. So it's 60, which does not tell you much, but this allows a certain breathing through the mask. And as I said, in parallel, we helped different industries to develop these masks according to the recommendations of the Science Task Force. And very soon, we could then start also with the help of Innosuisse, we could start a large project in collaboration with the two associations, Swiss Textiles and Swiss Mem, where we started developing a very innovative technologies for masks that could also guarantee a certain long-term mask production in Switzerland. We are quite aware from these 50 companies that are now collaborating with us that only a few will probably continue with the production of masks, but we have around 5 to 8 partners that really are very interested to also have a long-term strategy in the development of masks. So from the academic side, we are the 4 partners from the 2 ETH and Laboratories. And so we develop different technologies on one hand to optimize the balance between filtration efficiency and air permeability, but also to be able to predict from let's say the textile parameters, from the diameter of the yarns or the fibers used, what could be the filtration performance. So we developed here with fluid dynamics, we develop for the moment a predictive model. And we also developed some new analytics, especially here on some structures that mimic the virus to understand the kinetics, more or less, of the neutralization of the virus. And with this, let's say one of the biggest innovation is really the development of alternative mask systems with the new membranes that we're developing, and also with some finishing like antiviral coatings. So when we come to this new analytics, of course, we want to look at the efficiency, for instance, of the antiviral coatings. So we cannot directly make all the tests with the actual virus, although our partner Laboratories has the possibility to test the surfaces with the actual virus, but on a daily basis, and to be faster in the developments, it's better to use an inactivated virus. And so there are some commercial products like this virus that corresponds to more or less an influenza virus. It's also commercially available here. And so with this inactivated virus, we have more or less the same geometry as the actual virus. So we have the same size and also the same surface charge as SARS-CoV-2. And with this, of course, it's safe to be used, and it's also easy to use. We are developing these analytics, so now we are considering also to use another type of coronavirus that is found in mice. So it's a murine coronavirus that will be able to help us to refine our analytics. So of course, the limitations for the moment is that this is not the actual virus, the SARS-CoV-2, and so the kinetics of the integration, for instance, of the capsules or the lipidic shell is we have to look if this is comparable to the actual COVID-19 virus. So we have to validate these methods, and that is done in close relationship with Laboratories. But we could already perform some preliminary studies, like here the effect of temperature or the use of detergent on the neutralization of the virus. And we see here from very preliminary results that when we look at 2 different temperatures and the use of 2 different detergents, we also always compare to a positive or negative control. So maybe I have to say that these virus mimicking structures, they have fluorescent markers, so we can look at the intensity of the fluorescence. And we see, for instance, here in these preliminary results that the detergent, here when we compare to the positive control, the detergent seems to play a more important role than the temperature in the deactivation of the virus. So for the moment, in the recommendation of the Science Task Force, we defined to wash the masks at 60 degrees with proper detergent. So if possible, detergent that contains bleaching agents, because this also promotes more or less the activation of the virus. But from these preliminary results, it looks like a lower temperature but with the use of proper detergent, could also neutralize the virus. So if when we look here at these two lines, they are superposed, the negative control but also the use of no detergent. So if we just rinse at 30 degrees, this will not deactivate at least this virus mimicking structure. It will not destroy it. So we now see that the detergents seem really to play a determinant role. But as I said, these are only preliminary results. We also look, because next or apart from the porosity of a membrane, commercial products that are melt-spun polypropylene membranes, usually, they also have a surface charge. So they're charged, and with this we have an electrostatic filtering of the virus. So usually these membranes are positively charged. the viruses are, and also SARS-CoV-2 is negatively charged, and with this we can have an electrostatic interaction. But when we use these masks for a longer period of time, and even some medical doctors asked us if they could recycle these surgical masks. And what we see when we expose these masks to water for a certain time, also to some disinfectants, but also only to water vapor for a certain time, we see a certain degradation of this surface charge. So we see here in these experiments that were performed by ETH in Lausanne, that we also have a reduction of the filtration efficiency. So this means that we have to find ways to really keep this surface charge on the membrane of the mask. And the measurements made when exposed to only water vapor, so we exposed the masks for several days to water vapor, so to a relative humidity of 85%, and we saw also a slow degradation of this surface charge, but this degradation was not that quick. So after 8 hours of wearing, the surface charge was still on adequate level, let's say. So the degradation of the surface charge occurred within days and not within hours when exposed to water vapor. Then another topic is the development of this predictor, so this mask predictor. So what we here are mostly interested is to look at the influence of these two parameters of the air permeability and the filtration efficiency. And so we developed this fluid dynamics, so COMSOL-based model. It's a physics-based model where we look at the air transfer, but also the mass transport. And so we can also adapt different parameters here from the space, so aerosol size distribution, for instance, or also the volume in the room, etc. And with this, we intend to be able to make a certain prediction on the risk to get infected. And so we have these different parameters here that we can define. We would then define a sick person, a healthy person, wearing either a mask or no mask. Here we have this physical model, and by playing then with the different parameters, we can then make a prediction. So for the moment, we work with influenza as a virus, as it's not known for the moment the viral load required to really infect people with the SARS-CoV-2. But we can play around then with the different parameters, for instance, here playing with wearing a mask or wearing no mask here. So if both are wearing a mask, we see that the time to reach this ID 50, so that's the risk of 50% to get infected, would be at around 500 minutes. And if one of the two is not wearing, so if the sick person is wearing mask, but the healthy person not wearing mask, this falls down to 71 minutes. And where we can also play here is with the activity of the people. And here we see, depending on the activity, so we then, of course, breathe more heavily. We also see here a drop of this time to get infected to when there is very heavy breathing. When one is not wearing a mask, this would be reduced to 2 minutes. So with this, we can really then optimize these two parameters of the air permeability and the filtration efficiency of the membranes that we're developing. And so we're coming to the development of these membranes. So here a chart where we show all the measurements that were made from the different institutes, from medical type of masks and community masks. We see here the breathing resistance. So to remember, the threshold that we set is below 60 for the breathing resistance and for the filtration efficiency with particle size 1 micrometer. We set this threshold at 70. So we see here this category would be the mask that would correspond to the recommendation of the Science Task Force. We see that most of the masks, of the medical masks, really, even if the filtration efficiency is not measured with these particles, we see that most of the medical masks on the market in Switzerland fulfill these requirements, so have a filtration efficiency of 80 or even higher, and have breathing resistance. So type 1 type of mask, they have a requirement of lower than 40. So we see we have quite a large amount of medical masks that fulfill these requirements. But we also have masks that do not fulfill these requirements, as we had at the beginning of the pandemic some direct imports of masks that were declared as medical masks but did not fulfill the requirements of the European standards. And this explains why we have these masks, but they more or less disappeared from the market as time goes. And in the community mask, we also see we have a large spreading of the results. Of course, the textile manufacturers, they were not used to produce highly filtering products, so they started around here, but now many of them are able to produce masks that fulfill the recommendations here of the task force. And in membrane productions, what we are doing, we are developing nanofiber membranes, and these are here, these masks that are in this circle. So we see here that we have a new category of membranes where we can have a higher breathability, but still a very good filtration efficiency. And now we'll go in a bit in more details of this membrane development. So these are very tiny fibers that we produce. So these are so-called electrospun fibers. So we can produce them either on lab scale, so with just a needle that is put on the tension, and we have a polymer solution here that we inject, and we then have with electrostatic discharge, we will have a fiber that will be produced. And of course, depending on the parameters that we are putting into the system, we can then produce fibers that have different diameters and different morphologies. So the diameter of these fibers are typically between 50 nanometers to 1 micrometers. And so these fibers are then collected here on this collector and produce such a mesh. And depending on the number of layers of fibers that we put onto each other, we can then change the porosity and with this the filtration efficiency of these membranes. So here we see a picture of normal polyester fiber and these nanofibers that we put on top of such a woven structure. So we can directly more or less produce this membrane onto a normal textile. So this is on lab scale. Of course, on industrial scale, there are also some plants, like we have a pre-industrial plant in our site of St. Gallen, and we can produce a lot of fibers in the same time. So with this, we have a higher throughput than just with the lab scale. And as I said, if we choose the parameters in a very specific way, we can produce fibers that even have pores or even holes in the fibers. So we can go into nano filtration, so nanoporous filtration. And we do this by combining different solvents. So the polymer, here we use a chemical model for this. So the polymer is here represented by this sphere. So here, this was polycaprolactone, and we can then determine if a solvent would dissolve this polymer or not. So if the solvent like DCM here or chloroform is within this sphere, this means that the polymer will be dissolved by this solvent. And if we have here the solvent that is outside, this would be a non-solvent for this polymer. So if we mix then here, for instance, the polymer solution, so PCl dissolved in chloroform. If we add a part of DMSO, we will have a phase separation in the polymer, and with this we can then create these pores. So that's what we've done here quite systematically here by changing here the ratio between the solvent and the non-solvent. So in this case, between chloroform and DMSO. So here by increasing the amount of the non-solvent, we can then from very smooth fibers create some structures in the fibers up to create some holes then in the fiber. And also the relative humidity in the chamber has an influence. So the relative humidity will influence the evaporation rate of the solvent, and with this also the morphology of the fiber, because the polymers during the fiber formation will align. So depending on how long the solvent will need to evaporate, we can have different morphologies of the fibers. So when we have a high relative humidity, we will have microcondensation at the surface of the fiber and create a further phase separation. So we see if we have a low relative humidity, very smooth fibers, high relative humidity with these microcondensation. So the formation of so-called breath figures will then create these pores. And with the use of non-solvent, then these very large pores that will, of course, then influence this the filtration efficiency. And with this, we can also, by changing the surface morphology, we can also create some specific drug delivery systems. So we can incorporate some active agents. And if we have here no pores, we will have a slow release. And if we have pores, then this will then increase the release of the active compounds, just because we have another surface to volume ratio in these porous structures. And so we can play around with the different parameters. We can also make several encapsulations. So here we put some active lipidic structures that also contain some active agents that can be then maybe even swallowed by a patient at one point. The fiber would disintegrate, and so the active compound would maybe only be released in the intestines, so would not be digested by the stomach. Or we can also use capsules that we can then integrate into these fibers. So we have a multiple encapsulation system. We can also use some similarly responsive polymers, so to have an active release. So here we could incorporate into the membrane some active compounds, and if we use, for instance, a temperature responsive polymer, we can then make the system it would swell and then release then the active compound when it's above a certain temperature, and when we switch back, the release will stop. So we can really make an on and off switching of the release, for instance, also with pH, which is really relevant for wound healing. And so we can, as I said, by playing around with the evaporation rate, we can also influence the structure of the fiber. So either a longitudinal structure, so more a fibrillar structure of the fiber, or more transversal structure of the fiber. So it's more a lamellar structure. And this will change, on one hand, the stiffness of the fiber, but also the release kinetics of the fibers. And we can also combine different polymers. So here we have a core shell structure of polyurethane core and a PVDF shell. And with this, we can create fibers that have the elasticity of the polyurethane, but the surface structure of the PVDF. This is, for instance, relevant in tissue engineering to influence the protein absorption at the surface of these polymers. So we have no absorption thanks to the PVDF, but we have the elasticity of the polyurethane, so we can use this for the growth of specific cells. We can also functionalize these fibers to have some active compounds, and with this make some sensors. And what is the very big advantage of these systems is that we have a very large surface to volume ratio, so a very high surface for a very low mass, and with this we can make very sensitive systems. So you see here an example of a sensor that we sensed ammonia, and we can then sense down to the ppb region. So we have very highly sensitive sensors, and depending on what we want to sense, this can be reversible or not. So we can either have non-covalent interactions, or we can have some chemical reactions by biochemical reactions, and with this we can make, for instance, sensors for different biomarkers. So like here, some volatile amines that are relevant for exhalomics. So when we exhale some molecules, we can, for instance, by detecting some compounds, we can detect or predict some, for instance, some kidney problems, etc. Here, one example also of a project that we have with different partners, where we make a smart wound dressing. So here we sense two biomarkers. When a wound becomes chronic, we know that the pH remains elevated for a while, and also the glucose concentration will drop, because the bacteria usually feed on glucose. So we will have a reduction of the concentration of glucose. So by measuring these two biomarkers, we can, with maybe fluorescent markers or colorimetric markers, we can then detect a problem at the interface directly at the wound. So this allows then for the care personnel that they don't have to remove the wound dressing to check the wound. They directly have an information about how the wound is healing without having to remove this patch. And also, when adjusting these parameters of the fibers, we can even make membranes that are transparent. This is a collaboration that started around 4 years ago with ETH in Lausanne, and now we managed to make these transparent masks that have the filtration efficiency and the overall parameters and properties of a surgical mask. So EPFL now made a spin-off of these activities, and the production should start beginning of next year. So here again we used the adjustment of the parameters to really create fibers that have a very small diameter, and with this we can reduce the so-called Mie scattering. So here we played around with two polymers. On one hand we have chitosan, and here caprolactone, and by mixing the different polymers, we can on one hand increase the mechanical stability. So if we use only chitosan, the product is not that mechanically stable. It's quite difficult to spin these only chitosan fibers. So with a mix, we can increase the mechanical stability of the membrane, but still then have sufficient transparency. So here you see the transparency independency of the fiber diameters. If we have fiber diameters in the order of magnitude of the light wavelength, we will have a large scattering, so the membrane will become opaque. But if we manage to make fibers in the range of, let's say, 20 to 50 nanometers, we can then obtain this transparency. And of course, as I said, we can also produce different layers of fibers on top of each other. If we increase the number of layers, of course, we increase the protection, so the filtration efficiency. But on the other hand, we reduce the air permeability. And of course, we also reduce here the light transmittance. So we can really say, if we increase here, you see the spinning time. So this increase linearly, let's say the amount of polymer that we put on the membrane. We see here that we have nearly a linear reduction of the light transmittance, but the air permeability will be reduced quite dramatically. And for for a surgical mask, we have to have this air resistance that is above than these 60 Pascal per square centimeter. So we have to calculate this back, but this is in the order of magnitude around here. So that's around what we can reach. So a certain air permeability and still have transparency of around 70 to 80%. And so I'm at the end of this presentation. I would like to thank my group for all the work that has been done, and also, of course, our funding agencies, and thank you for your attention.